Shane. How you doing, man?
How are
Hello?
you? Had a good Thanksgiving?
Yeah. I don't really celebrate it, but I know
Yeah.
I went to his house. So, how about you?
Yeah, it was really good. I went back to see my family and my like little cousins and stuff and had some really good food.
Nice. Wait, where are you from?
New York? Where were
Sweet.
you from again?
I'm from Miami.
Oh, yeah, okay.
So wait, so you're not originally from New York? Like you go,
Yeah, I think I told you I have two other students from Miami and like three other students from Florida.
yeah. There's a
It's
lot of us, it seems.
yeah. Like, did you grow up in Miami?
Yeah, well, I was born in India, but I moved here when I was like 6, and I've
Yeah,
been here.
so your family and everything moved to Miami though?
Yeah, just my parents. Just my
Mm.
parents.
Do you go to like the University of Miami or something?
No, I went to UCF actually, Central Florida, in Orlando.
Got it.
Yeah.
Interesting. Yeah. It's like, you know, super warm there, I'm guessing. Or like I guess for you, like 70s would be considered cold, right?
Yeah, it's like 70 today, and I'm freezing.
Yeah.
I went to work, I went to research today, and I was like, damn, yeah, I should have brought the sweater today. But
That's funny. That's funny. Yeah,
but yeah.
student one of my students in Miami is like from New York originally and he just you know uh likes it a lot. But yeah, I went to Miami like once for spring break and it was pretty beautiful. So it's definitely like a paradise.
Yeah, it definitely is. But
Oh,
yes, since last time we saw each other, I ended up taking Blueprint 1. No, sorry, not Blueprint 1. Oh my God, Blueprint 7. I meant to say Blueprint 7.
yeah, now um if you can just do you know in your spreadsheet like the full lengths tab if you could just input your test scores there, that'll be
Mhm.
helpful.
Uh,
It's
yes.
the tab called
I'll just put for 1 because I don't, or I'll just change to 7. There we go.
because you what?
Yeah, I'm just because it was from 1 to the first one is 1, so I'll just change it to 7. Yeah, so I got for CP, I got 126. BB, I got 126. And then PS, I got 127.
Okay.
And then for cars, I got 36 out of 53 on the Jack Weston,
Okay.
but it seems it's a 125. So I don't know. What do you think?
I'm gonna use a.
What day was it? 3, 2, 1.
You said it was how many?
36.
Yep, 125.
Yeah, so it was and then I did the blind review. So something
Yeah.
it seems like the cars is definitely working out because it was crazy. I was pretty locked in for the first 5 passages, which I've been used to.
Yep.
And I ended up getting a 26 out of 30 on the first like 5 passages.
Mm-hmm.
And then that's and then that's when it just went downhill. My
Uh-huh.
stamina was like dying, but I
Okay.
re
So we can okay, let me insert that as a note here. So doing great
oh, sorry, I'll wait for you.
for the first how many?
Like the first 30 questions, I got 26 out of 30. So like first 5 passages, I think.
Oh, 1 second. Hold on.
First time? What day was it? December 12th.
Sorry.
Oh,
Uh continue.
yeah. So
So it was
So yeah, the first 30 I did really good.
Yeah, and
Just
then
went for
uh
stamina
yeah,
issues. I was like,
yeah.
the timing was short. Then I was like, oh shit. And then I kind of just, it
Uh-huh.
just plummeted after that. But what I did was I just kind of guessed on the last, like the last two passages I didn't even see. And then I was barely reading the last like four. And what I did was I did the blind review, but I didn't do the beginning because I was like, okay, I feel good about it. I'm just kind of
Yeah,
wasting time here.
sure.
And I got like a 20 out of 23 on the second half of it.
Got
So yeah, that was, well, not 20 out of 20. I think it was like, yeah, it was like around 20. Yeah. Like overall, I got, if timing wasn't an issue, I probably would have gotten at least 45 plus, which was insane to me.
yeah.
So it definitely seems, I think going forward, I'm not gonna like put timing into it. I'm still gonna continue to build myself to 9 passages, then I'll put timing in. But yeah, that was exciting. That was honestly
Yeah,
really
yeah.
exciting to see. So
So tell me a little bit more. Like, so were you using like the cart, like the mapping strategy? Like what were you?
um I realized I shouldn't like it takes me a little bit too much with the mapping.
Yep,
So like I
yep.
kind of went along with what you were saying and I kind of started to get it was the passages seemed a little bit easier to be honest
Yeah.
with you. Um so I think I wasn't really mapping it out, but I really made sure at the end I still understood the main idea.
Yeah.
And that's like the biggest thing that I'm still trying to focus on.
Yeah, yeah, yeah.
But yeah, mapping, I kind of like for the first two, three, I kind of write whatever, but then
Hmm.
I just keep going on from there.
Yeah.
But
Yeah. So
yeah.
that's absolutely great. Because yeah, as long as you so I'm guessing like what you're doing is like you're reading it and you're kind of, you're building the skills to like actively read, to think about what you're reading as you read it.
Hmm.
And um you're also spending, I guess, like a couple of seconds after the passage to kind of think about the main idea, anything like that.
Not specifically, I just think about what I just wrote. I'm like, okay, like what did I just write? Like when we're not right, what did I just read?
Uh-huh.
Just like processing what it said there. And I kind of just kind of like made it's been easier to make a little bit of a mental note.
Yep, yep.
Um, but yeah, like I haven't been writing out because I realized after a conversation last time
Yeah.
that I'm still like beforehand, I'm still focusing on like rewording the thing and not really
Yep,
like
yep.
understand the argument.
Got it.
So yeah, so I just
Okay.
tried to start making a mental note of it. There's like certain like structure aspects that's still kind of an issue, and I see myself like going back to those parts and like
When
kind
you
of
say
rereading
structure
there.
aspects, are you talking about like the structure questions?
Yeah, like go back to paragraph 4 for a question.
Yeah,
So
that's fine. That's fine for those.
okay, okay.
Like the when we do it, like that's when we do it with a full map. So, if you're doing what you're doing, which means like you're not writing out the full map, then yeah, if you have to go back, you'll go back to the passage. But as long as, as long as like that's the only time when you go back. Like if they're asking you any other question, you should have, if you have a good understanding of the main idea, you should be able to answer most of the questions without having to go back. So as long as it's not like a reflex, because for most people when they start out, it's a reflex to read the question stem and then go back to the passage no matter what. So for you,
Mm.
if you're saying that you're not doing that, you only go back if it's a structure question, then that's good. Is
Yeah,
that accurate though? Like,
yeah, typically like even when I was like doing the mapping and stuff, like as I got a little bit better, like I didn't even go back to my map typically.
yeah,
So
yeah,
I kind of just like, I think this seems pretty right. So I
yeah.
just kept going.
So
Um,
so would you say that you look back at the passage predominantly for the structure questions, but not for the like main idea questions, the other questions?
yeah, I don't really think I look back on the main idea.
Okay.
I kind of got a
Good,
knack
good,
for it.
good. All right. That's good progress
Yeah.
then.
There might have been certain points that I looked back, but
Yeah.
overall it was kind of
As long as it's not your reflex, right?
Mhm.
Um, okay, that's really good news then.
Yeah.
Well,
But
good.
yeah, so I think there still need to be work, but I think that's a good starting point, better than the 120s I was getting before.
Yeah,
So
now,
yeah.
you took some other tests recently, right?
Other
Oh, you
tests?
no, you didn't then.
No, this is my first full length I've taken with you.
Okay, okay.
I mean,
So,
diagnostic, I don't know if you want to count that as a test, but
uh-huh. And
then no.
okay, good, good, good. This is a good place to start then. All
Okay.
right. Yeah, what would you like to go over today?
Okay. Um, yeah, so I guess we can go over like BB stuff. I did the plan reviews on those too. So again, I think it's a mix. I think a lot of there were like very like a couple content stuff, but a lot of them seem to be the passages. Yeah, it's been so long since I've been doing passages, so it just seemed I
Mhm.
felt I was getting very like tired and out of focus a lot, but somehow I was able to do decent. So yeah, I'm not showing my screen. I guess we can work on this one. This question. Yeah, the questions didn't seem as terrible, but I feel like the passage, like if we were to go through this passage, I think that might be helpful in kind of like putting together all like the metabolic pathways and stuff and like
Mhm.
and reviewing that. So I guess that this would be a good starting point.
All right, let's zoom out a bit.
Zoom out. Yeah.
See if you could do it a little bit more, like zoom out.
Zoom out.
Yeah,
Oh,
you see the mic go. Oh, yeah.
more?
Oh, this is good.
Okay.
So, oh, so on the left, to fix that, you can just put your cursor here and like drag it down and then drag it back up.
Oh, nice.
So, did I show you like the proportionality type of thing?
Yeah, we didn't get to that. We were only doing CP last time, so
Got it, got it. Okay, so this will be a good one then. So, um, we talk about buzzwords though, right?
yeah, but last time when we went over, I feel like there didn't seem like many of those buzzwords, so I feel like we didn't really
Yeah, yeah,
go.
yeah. Okay, okay. But yeah, buzzwords are words that you are expected to know about and therefore likely for them to ask questions on.
Yeah. Okay. Hmm. Did you ask me something?
Hmm?
What did you ask me? Sorry.
Oh, so yeah, so you could just read out loud slowly.
Okay. Um, Oxidation of long chain fatty acids involves formation of an acylcarnitine molecule in the cytosol by the transfer of the acyl group of a long chain fatty acyl-CoA molecule from coenzyme A to carnitine.
Okay, so already a lot of stuff going on here.
Yeah.
So, what can I guess, yeah, what can we say about this?
Yes, I think it's basically talking about like beta oxidation, and what really kickstarts it is the addition of sort of this acyl group from coenzyme A
Yeah.
to carnitine. Seems to be the big thing here.
Got it. So, um,
Oh, I wish I could take out my highlights.
oh no, that's fine. So, okay, so oxidation of long chain fatty acids forms acylcarnitine through this transfer of an acyl group of a long chain fatty acyl CoA from CoA, coenzyme A, to carnitine. So let me see how I may write this. So maybe I would put um, LCFAs through oxidation forms acyl carnitine through the transfer of an acyl group from a long chain fatty acyl CoA. I'm trying to think if that long chain fatty acyl CoA is the long chain fatty acid, but whatever it is, we're taking
So
uh, the seal from the CoA to give to the carnitine. So maybe I'll just put like um, CoA and then maybe a seal and then here just CoA. So the acyl has added to the carnitine. Okay.
coacyl. Okay. Oh, so it's not just acetyl CoA. It's a long chain. I see. Okay. I see what you're saying. So the acetyl CoA is part of some sort of longer chain, and then that's what transfers it. Okay. This permits fatty acids to be transported into the mitochondrial matrix in the form of acylcarnitine. Okay. For breakdown to acetyl-CoA in the beta-oxidation pathway and subsequent oxidation in the TCA cycle.
Okay. So how well do you know like the beta oxidation pathway?
To be honest, yeah, it's been a while.
Uh-huh.
Um, I know it goes to the carnitine shuttle thing. That's
Yeah.
besides that, it's kind of like
Okay, okay. Because
much.
so typically what I would write for these types of passages are the thing that you saw me write or draw.
Yeah.
But I realized that there's a lot of that happening in this paragraph, and it won't be the like I don't think it's going to be the best use of our time by writing out all of that stuff. So, but if we recognize it as beta oxidation, that could
Mm.
kind of help us out in terms of, even if we don't know the specifics. Well, I think they're telling us the specifics, but as long as we know like generally what beta oxidation does, right? And it's the breakdown of fatty acids, right?
Yeah, into acetyl-CoA, right?
Mm-hmm. So, okay, so let's just take that away from this first paragraph. All right, you can continue.
To investigate a possible relationship between changes in the oxidation of long-chain fatty acids and insulin resistance in mice, or in liver cells, sorry. Mice were fed a high-fat diet for 8, 16, or 32 weeks. So it seems like they're checking for changes in beta oxidation and how it might relate to insulin resistance.
Okay.
Mice on a HFD were found to have high levels of acylcarnitine, a finding consistent with the elevated beta oxidation measured previously in insulin-resistant muscle. Okay.
Okay. So it seems like so I'm actually gonna get rid of this. I'm gonna write a proportionality here. So acyl carnitine is proportional, or I'll put like this to mean like concentration, is proportional to beta oxidation, meaning higher amounts of acyl carnitine indicate higher amounts of beta oxidation, right?
Yeah.
Okay.
When challenged by insulin injection, data confirmed similar changes in hepatic insulin sensitivity fed for mice fed in high HFD for 32 weeks, impaired insulin signaling, increased hepatic gluconeogenesis, and a decreased fraction of hepatic glucose production due to non-gluconeogenic sources in these mice versus age-matched controls.
Okay. So,
One second. Sorry. Keep getting these notifications.
mhm.
All right.
So okay, yeah, what are we saying here?
Um, yeah, this part confused me when I was reading it, but it seems like when they tried to use insulin on these mice that were fed a high-fat diet, it seems like it caused impaired insulin signaling and increased hepatic. Well, actually, no. Well, I guess when they used insulin when they gave insulin to these mice, it seems that there were changes in the hepatic or the liver insulin sensitivity. It seems.
Say that part again.
Yeah. That when they tried to put insulin in these high-fat diet, like in these mice, it seems that it caused changes in the insulin sensitivity of these hepatic cells.
So does it say that it causes the changes?
Hmm.
Or,
Just similar changes, so I guess not.
well, there's a different word there.
Confirmed similar changes?
Mm-hmm.
Confirmed. So,
Yep. So, okay, so it seems like prior the high levels of acetylcarnitine are consistent with high levels of beta oxidation. And then they said measured previously in insulin resistant muscle. So I added that to my proportionality here. In parentheses, I put insulin resistant to mean this is what happened, that this proportionality relationship happens in muscle that is insulin resistant, right?
okay.
So then what they said was when challenged by insulin injection, the data that they obtained confirmed similar changes in hepatic insulin sensitivity for mice, fed the high fat diet for 32 weeks. So it's when you inject insulin into these, you know, mice that we saw like that high level of beta oxidation for, we saw that there's that that it confirmed that there were changes in insulin sensitivity. And then we have a colon saying that these are perhaps the changes. So impaired, so like examples of the changes of insulin sensitivity. So impaired insulin
Mm.
signaling, increased hepatic gluconeogenesis, and decreased fraction of hepatic um, glucose production due to non-gluconeogenic sources in these mice versus age-matched controls. What are they trying to tell us here?
That I guess insulin seems to not be doing its effect.
Tell me more.
And specifically, we can see how insulin is not doing its effect, as you said, because of the impaired insulin signaling, increased hepatic gluconeogenesis. Because typically, if insulin, typically with something like if they injected insulin, gluconeogenesis will probably go down because you already have enough, because you want the cells to take in glucose.
Yeah, you want to store it, right? You
Yeah.
don't want to make more of it.
Hmm. And then decreased, that part I don't get. Decreased fraction of hepatic glucose production due to non-gluconeogenic sources. I'm not sure how insulin would affect that for some reason.
So maybe you're only seeing the part where it says decreased fraction of, well, so okay, tell me what you're thinking.
About the decreased fraction part?
Yeah.
due to non-gluconeogenic sources. Um, so I guess in a normal condition, in like the age match controls, like insulin would impact or increase non-gluconeogenic sources.
Yeah, so um the glucose that gets created that comes from a non-gluconeogenic source would be something like maybe from a fat, from a protein,
Hmm.
something like that, right?
Yeah.
So there's less of that happening. So, okay. Um,
Okay, I see what you're saying.
yeah. Okay.
So nothing from fats, nothing from like fatty acids and stuff, nothing from proteins. Okay, that makes more sense. To confirm changes in fatty acid oxidation and TCA cycle flux, the rate of oxidation of the labeled fatty acid 114 carbon palmitate in liver homogenates after 16 weeks of HFD was examined for 14 carbon dioxide production.
All right.
Mhm. Seems not too bad. Okay.
So,
Isolated.
well, we want to look at the figure.
Oh, right. Yeah, yeah. So compared to the control, we see that the high fat diet, there's more carbon dioxide production. Um, so it probably means that yeah, they're forming. Well, I mean, from what I know, and after reviewing this, it seems like the palmitate is being broken down, and the acetyl-CoA is being thrown into the TCA cycle, and
Okay.
then through the TCA cycle, like I think the formation of like alpha ketoglutarate and succinyl-CoA releases, or no, sorry, like isocitrate and that part, somewhere along those lines, there's like a release of carbon dioxide. So I
Yep.
guess there's continuous increase of that.
Yeah, right. So if your metabolic pathway is produced, because all in all, like in cellular respiration, right? It's like some hydrocarbon, like a sugar or glue or something like that, and then like oxygen yields CO2 and water, right?
Mm.
So measuring the CO2 production can give you an idea of the metabolic rate of something,
Okay.
right? And
Yeah,
so we
good.
see that for the high fat diet, um, and these are for the regular mice, okay, or yeah, regular mice.
I think the
Mice
white is,
fed the high fat diet.
yeah, the white's control, and then I think this is the high fat diet.
Oh, oh. So, and the control is just a regular diet?
That
I'm
they
trying
didn't.
to trying to see if there's any type of mouse that has like insulin resistance or not.
No, I think the only thing is like they were just fed a high fat diet.
Okay.
So I think compared maybe yeah, I feel like you're right. I feel like it's probably just a normal diet. Or maybe
Okay.
not.
So here we can just see that high fat diet proportional to CO2 production.
Okay.
Okay.
Isolated mitochondrial respiration in those homogenates was measured as oxygen consumption with palmitate-derived palmitoylcarnitine and malate as substrates under either ADP stimulated or ADP or ADP simulated 1 micromolar of ADP, state 1, or ADP depleted 0.1 micromolar ADP, state 2, conditions in mice fed and high-fat diet for 8, 16, and 32 weeks. So it seems here they're moving on to, yeah, so measuring oxygen consumption, but using this as substrates, some sort of this whole long thing, and then this as substrates in these sort of conditions. And they're also fed high fat diets, it seems. Okay. A respiratory control ratio, RCR, was also calculated as the ratio of mitochondrial ATP formation to oxygen consumption. Each value was expressed as a percent difference from respective age-matched control mice fed a normal diet.
Okay. So,
Okay.
yeah.
Interesting. Okay. This graph is a little weird to understand here, but difference from control. Yeah, this is a part I don't, I'm not sure about the O2 consumption because it's rating this, but I'm guessing, wait a second. I'm
What-
guessing increased levels of this difference would indicate possibly higher O2 consumption. They don't explicitly say, but that's what I'm thinking.
Let's see, right? So we have our y-axis of the palmitoyl carnitine and malate substrates that can produce um let's see, an RCR, which is the rate of mitochondrial ATP formation to O2 consumption, and that each value is expressed as a percent difference from respective age-matched control mice fed a normal diet. So it seems like, and then we have state 1, which has a lot of ADP, and state 2, which has a small amount of ADP. And so, yeah, so it seems like for state 1, so no matter what, for state 1, oh, and then RC, oh, okay, hold on. RCR is like has its own column. So I thought that was going to be the y-axis, but let's see. Each value, percent difference from control. Okay, y-axis is different from control, aka mice fed a normal diet, right? Like
Okay.
that's what the y-axis means. Meaning,
There's 40% difference compared to control of this substrate, I'm guessing.
uh-huh.
Okay.
Compared to the control, uh which has been fed a normal diet,
Yeah.
right? Okay, and then we can see in state 2, when ADP is depleted, we have for the 8 weeks a more negative percent difference. For the 16 weeks, it's pretty much the same. For the 32 weeks, it jumps up even higher. And then, so it seems like, let's see, it
Is
seems
it
like,
is it consuming more oxygen because it wants to make more ATP because there's not enough ADP, or that's what I'm thinking.
so,
If it's depleted.
so yeah, when ADP is depleted, state 2, it seems like we have this higher amount of difference from control, ADP, higher perhaps difference from control. And then for the RCR, oh wait, sorry, ADP depleted. So this should be like 1 over ADP. It's an inverse proportion. If you have less ADP, you have a larger difference uh from the control, right?
Okay.
And then we have the RCR one, which is the ratio of ATP formation to O2 consumption. And we can see that it's the 8-week bar that has a positive, difference. Whereas the 16-week and 32-week ones, well, the 16-week one is lower and the 32-week one is, in the negatives compared to control. So it seems like as time passes, we have a smaller amount of mitochondrial ATP formation, right?
Mhm.
Because RCR is the ratio of the mitochondrial ATP production to O2 consumption, right?
Yeah, it's negative for those become more negative.
Mm-hmm. Okay. Anything else on this passage?
No, that was it.
All right. So, not the easiest, and we still just have pretty much like a cursory understanding of what the figures mean, but that's okay. Let's move forward. So, yeah, why don't you try this question out?
Okay, so based on the findings presented in the passage, impaired insulin signaling, impaired insulin, impaired insulin signaling in mice is marked by, as far as I know, again, as we talked about, it seemed that it was the insulin injection caused this insulin sensitivity,
Yeah. So,
changes in
impaired insulin sensitivity is marked by a decreased what, right? So, it says increased hepatic gluconeogenesis, right? So that so the total so A total hepatic gluconeogenesis should increase, right?
Okay, from what they say. Okay.
And then let's see, um, decreased fraction of hepatic glucose production due to non-gluconeogenic um sources, right? So we should have a well, this is tricky, dude. So because it says decreased fraction of hepatic glucose production due to non-gluconeogenic sources. And then B says non-gluconeogenic glucose production as well. The only thing that could be an issue is fraction. Um, let's see. While the passage does mention that these cells displayed a decreased fraction of hepatic glucose production due to non-gluconeogenic sources, this may be attributable to an increase in the rate of gluconeogenesis and is not necessarily a consequence of decreased glucose production from other sources. Hmm.
Yeah, I tried to reread that
So
5 times. I still didn't get it. I was like, whatever.
yeah, 'cause I thought, well, when I first read it, I thought impaired insulin signaling would be distinct from increased hepatic gluconeogenesis, and that to be distinct from decreased fraction. So, but they're saying that this part is consistent. Can you show the explanation for A?
Yeah. There you go.
Impaired insulin signaling cells that also show. Well, I mean, okay. And then let me see. The hepatic insulin sensitivity. When challenged by insulin injection, mice fed, okay, confirm changes in fatty acid oxidation and TCA cycle flux, the rate of oxidation of the labeled fatty acid palmate, okay. Passage further states that these findings confirmed a change in insulin sensitivity that was similar to that seen in insulin-resistant muscle cells. Taken together, these suggest that the liver cells of the mice tested likely displayed both an impairment in insulin signaling and a decreased. I guess that if we take that entire second paragraph into consideration, they say that um, that they are using mice that have insulin resistant muscle. Hmm. And then, okay, when challenged by insulin injection, data confirmed similar changes in hepatic insulin sensitivity. So,
Yeah, because all they say is similar changes, though. Can we say that it decreases?
mhm. Okay, I think I understand. So, they're asking us what impaired insulin signaling can cause. So, it can cause, like, when insulin binds to an insulin receptor, it's not going to work very well, and increased um, hepatic it can lead to increased hepatic gluconeogenesis, um, because if the liver has impaired insulin sensitivity, it's not going to be able to bind, or the insulin's not going to be able to bind to the receptors of the cells of the liver. And if it can't do that, it means that there's going to be an excess amount of blood glucose, right?
Yeah, that makes sense.
And now, if there's an increase in the amount of blood glucose, there shouldn't be any, let's see, Yeah, so I think that for this question, it's just that impaired insulin signaling is most directly um more directly connected to, I mean, if you have an issue with insulin signaling, right, you should have a decrease in insulin sensitivity, right?
Yeah, that makes
That,
sense.
yeah,
But
but
the thing is, like, it just the way they word it too in the
yeah,
beginning,
I
because it was
know,
like
I know.
baseline findings. Just
Yeah,
like,
yeah,
I don't know.
yeah. Yeah, no, the way that it's worded makes, I mean, like, I would be fine with this, but if they're saying that this part is true, but this part isn't, that's where it gets confusing. So, yeah, I'm with you on this one, and most people are as well. More people chose B than D. So I think that just the most direct answer would be because when you think about insulin sensitivity, like, sorry, when you think about someone's like with diabetes, right?
Mhm.
There's impaired insulin sensitivity, and
Mhm.
there could be these downstream effects, like some with hepatic gluconeogenesis or hepatic, glucose production from non-gluconeogenic sources, right? Those could be like downstream effects, but it initially all starts with that in, uh, decreased insulin sensitivity.
Okay.
So,
No, I definitely see what you're saying, but yeah, I don't know. It's just the
yep, yep,
deal,
yep.
the part
Yeah,
just
I'm with you on
whatever.
there.
Okay. Um, this I understood. So I have to focus on this. Then we can do this one.
The amount of CO2 produced in ADP stimulated mitochondria that oxidize this thing is okay. So we're gonna look at our figure two, right?
Hmm.
And where are we gonna where I guess are we gonna look?
I guess basically between state one and two, right? So
Uh-huh.
stimulated. So
The gray bar, right, for state
yeah,
one
so six, yeah,
is
you're right. 16 weeks, right? So yeah, it's the gray bar.
okay.
Yeah, so it seems the gray bar here, they're pretty much constant. Not constant, but they're the same level. Sorry, that was a better way to put it. Same level
Yep.
of the difference from the control.
Uh-huh.
Okay, so I put decreased. I don't even know why I put that, but
Decreased more versus controls than
I think I might have been looking at the black bar or something. I don't really remember. Um,
that should still be increased though, right?
hmm,
Maybe you're looking at the white bar
no,
or
I think maybe I made a mistake because instead of 16 weeks, I might have been looking at the 32 weeks for some reason, which is the black bar. But
uh-huh. But
wouldn't that be decreased though, right? If let's say if it's at 32 weeks here, comparatively, state one is the stimulated one
mhm.
versus state two, the ADP depleted, right? So
Yep.
wouldn't it mean that there's decreased levels?
Yeah.
Okay.
If they said yeah, so B, I believe,
If it's at
would
32
be
weeks.
correct if they said 32 weeks.
Okay.
So,
But this makes sense though, because it says increased equally versus controls, because they're essentially the same level. So we can kind of say that they did.
yep. Mhm.
I think maybe when I was doing this, I feel like the CO2 definitely threw me off because I was like, they only talk about CO2 earlier here, but this I wasn't able to make the link there for some reason. I think that kind of just threw me off.
Oh,
But it just makes sense, right? Because like these substrates are being used through the TCA cycle and then eventually to the electron transport chain. Um, so I guess they're trying to talk about that, right? The CO2, like the CO2, like the more difference there is, the more of them are being used into the TCA cycle, right?
yeah, yeah. Yeah. So like in general, like CO2 production and O2 consumption give you information about cellular respiration.
Okay.
Like, I forgot what the test is called, but like, have you ever had like your metabolism measured?
Yeah, they poked a little thing in you, right?
What? What thing?
They poke something into
Are
your
you thinking
finger.
about like checking like
But
your blood sugar?
yeah,
Oh
is
no,
that
I
what
mean
it,
your metabolism.
I don't think so, no.
So so okay, so everything goes like ultimately still goes through the Krebs cycle and electron transfer chain, right?
Yeah.
Like even proteins and fats, they break up into like acetyl-CoA and they still go through that, right?
Yeah.
So cellular respiration is, typically glucose, but it could be any type of hydrocarbon plus O2 yields water and CO2.
Water and CO2. Okay.
So if you, so like, when I had my metabolism checked, like they give you like some mass to just like breathe into and like also like exhale. And so it can measure the rate of your oxygen consumption and your CO2 uh, production to figure
Mm.
out what your metabolic rate is.
Okay.
And also, interestingly enough, like mostly all of like the carbon in the food that we eat, it doesn't exit as like, feces or anything. Most of it exits as CO2 that you know, exhale.
Mm.
It's like, have you ever like weighed yourself at night and then when you wake up, you weigh yourself?
Yeah, I think I'm typically less right in the morning.
Yeah. Now, how could that be? Because you didn't like have a bowel movement, right?
Yeah.
So it's funny. There's like Louis C.K. or whatever. He's a comedian. And he was talking. He did a bit once where he's like, "Yeah, I measure my weight before I go to bed, and then I measure it after, and then when I measure it after, it's smaller. So that means that I, take a, shit in my bed like every night." But no, yeah, it's because like yeah, I always wondered that like how where did that go, right? I didn't like
Mm.
excrete it or anything. And the answer is that it goes as CO2. Like that carbon, because yeah, think about it, right? C6H12O6 plus O2 yields CO2 plus water.
Mm.
So like these carbons can only leave as carbon and carbon dioxide.
Mm.
Yep.
Through all those like the oxidation and all that stuff. Okay.
Mhm. Yep. And also, this is a combustion reaction. A combustion reaction is when you take something like oxygen
Mm.
and you let something combust, like breaking it apart. Um, if you replace this glucose with a different hydrocarbon like octane or like petroleum, like that's gonna be like it'll still be a combustion reaction, but it'll be like one for like a car. That's why a car uses an internal combustion engine. So
Mm.
it's the same type
Okay.
of yeah, same type of reaction. So yep.
Okay. I'm gonna screenshot some of this.
Mhm.
Let's keep going. Um, I think we can go to there's this passage here that was really seemed tough. Can I use the bathroom real quick?
Yep, go ahead.
Yeah, so yeah, this passage felt pretty tough. I know I got a couple of them right, but honestly, it just felt kind of like lucky guess. So,
Seems like a lot's going on here. All right, you can start reading it.
okay, so in most animals, mitochondrial DNA is inherited maternally, but another mode of inheritance known as doubly unilateral uniparental inheritance, DUI, occurs in certain species of bivalves. Don't really know what bivalves are, but it seems like mitochondria, yeah, it goes to your mom, but in this place it goes through some inheritance pattern, another mode. DUI involves sex-selective heteroplasmy of two highly divergent and perhaps evolutionarily distinct genomes known as F and M mitochondrial genomes, female and male.
What do you think this DUI thing means?
Um, that maybe it says doubly uniparental inheritance, so just through one parent, like it doesn't matter if they're male or female, it just gets inherited through one parent. That's what I'm thinking.
Yeah, and you could even use like a cars trick here, like this. If we're saying mitochondrial DNA, like in most animals, is inherited maternally, but we already know that what's after that contrast were to likely say something like, but it could also be done like passed down paternally.
Mm, okay, I see what you're saying.
So, and then they throw this like big word at you that's meant to confuse you, but you know that that could only mean one thing, that it can also be passed for the from the father.
Okay.
And I
Yeah.
believe a bivalve is like a clam.
Mm, okay.
So, oh, and then they said, so DUI involves sex selective heteroplasmy of two highly divergent and perhaps evolutionarily distinct genomes known as the F female and M male mitochondrial genomes. So, um, what do you think they mean when they say like, divergent and evolutionarily distinct?
I mean, the most I know about divergent evolution is when you have like one species and they essentially share the ancestor, but they split off and continue on their own, like a different path.
Uh-huh. Yep.
Uh,
So, uh-huh.
Evolutionarily distinct, so I guess yeah, that kind of goes hand in hand, I guess.
Yeah,
That
so the F mitochondrial genome and the M mitochondrial genome likely come from like different sources, evolutionarily speaking, right?
okay.
Okay.
The F genome, which is localized to the gonads, is transmitted only by females to offspring of both sexes. Whereas the M genome, which is normally found in the mantle, undergoes only male to male transmission. Okay,
Okay. He thought you like female can go to male or female, but male can only go to male, right?
yeah, male to male transmission.
Okay.
Okay. Consequently, females are homoplasmic for the F genome, while males display heteroplasmy, meaning that they simultaneously contain both F and M genes.
Okay.
So it
So
seems
now,
that the F genome can go to both, but for some reason they only have the F genome.
So yeah, so females have the F genome, and males have
F plus M.
F and yep, plus M. Okay, like that.
Mm.
And like this isn't even like really like a schematic, but it's like, it helps. I think it's always useful to like be able to visualize this stuff.
Mhm.
So just helps you visualize it. Yep.
Okay. A study was performed to determine whether the diploid mitochondrial genome of the muscle per purpuratus display the sex-selective heteroplasmy characteristic of DUI. So essentially checking the mitochondrial genome of the species to see if this heteroplasmy stays true or those of this DUI. Samples of tissue from the mantle and gonads of mature adult purpuratus, I guess, were collected. Total DNA was extracted from the samples, and the cytochrome oxidase subunit 1, CO1, mitochondrial gene, and 16S region of mtDNA was amplified. Okay,
A lot going on here. Let's see.
so trying to check, they have a mantle, some sort of tissue that doesn't seem too important, but the last part seems to be important. They're checking for COI, mitochondrial gene, and 16S region of mtDNA were amplified.
Uh, okay, so yeah, they're trying to figure out if the diploid mitochondrial genome of this muscle displays the sex selective heteroplasmy characteristic of DUI, aka female has just F genome and males have F and M genomes. So they took samples from the mantle and gonad. So like from the mantle, like that's gonna be from the male, gonad from the female. And they collected them and they took their DNA and they amplified the cytochrome oxidase subunit 1 mitochondrial gene, which I believe is like in that ETC, right?
Cytochrome oxidase, yeah, complex 4, I think.
Okay. Um, and this other region. Okay.
The number of polymorphic sites and the number of haplotypes. Polymorphic sites known to commonly be inherited together. Present in samples collected from 10 females and 10 males were estimated for COI and shown in table 1. So they're essentially checking on these genes for polymorphic sites, which are, I think those are regions with a lot of the same repeating units, right? And they're just checking if both
What
of them are
what was
present. Polymorphic sites are essentially just regions with repeating nucleotides, right? I think.
not sure. Um, but they do tell us that haplotype what are haplotypes?
To be honest, not sure.
I mean, from the passage?
I guess the F and M genes, I guess. Genomes, haplotypes.
Well, what does it say?
Oh, polymorphic sites known to be commonly, sorry, polymorphic sites known to commonly be inherited together.
Yep. So that could be what a haplotype is, right?
Oh, okay. Okay. We have number of sequences for the F genome, so we have 30. For the male, we have 10. And then female, there's 20 sequences. And then for male, there's 20 sequences. The sequence length is the same, everything. Polymorphic sites, 27 in F genome, 18 in M, 21 in female, and 25 in male. And the number of haplotypes, which are again polymorphic sites that are inherited together, there's more of them for F versus M, but more in the male versus the female. F genome versus them. Yeah. Okay.
Yeah. Hmm.
Weird stuff.
Yeah. Um, okay.
So it says indices of genetic variability of COI sequences are organized separately according to F and M genome source and female and male. Oh, female and male gonadic and somatic tissues together. Source and P. purpuratus. Okay. To detect differences in the number of mutations, so the number of mutations accumulated over time between sample types, and thus to establish if an apparent difference exists in the age of each genome. An analysis of expected versus observed mutation, sorry, frequency was also conducted. So they wanted to check for the amount of mutations that were happening over time. Uh, it seems that there were more mutations in the F genome comparatively than the M, at least in this distance. Okay. And that is it.
Okay.
Explain to the questions. Do the estimates shown in table 1 support the conclusion that certain mitochondria of P. purpuratus are heteroplasmic? And when they said that heteroplasmy was they simultaneously contain both F and M genomes, while males display heteroplasmy, contain both F and M genomes. Yeah, like this, I don't, we know it's what, 10 males and 10 females and 10 males. I
So,
genuinely don't, I don't know how I got this right.
so heteroplasmic we know means, for instance, that, well, for the male, that they have both the F and M genomes.
Yeah.
And let's see. So I think I'm not sure if I should look at number of sequences or not, but so if I'm comparing F genome and M genome, the sequence length is the same. The number of polymorphic sites is slightly different. Um, Number of haplotypes, tiny bit different. Number of sequences is the largest difference. Now, okay, now if they're saying that we have an equal number of females and males, meaning 10 females, 10 males, and that the number of sequences for this COI gene is uh well 20 for the male and 20 for the female. Yet we seem to have so we have an equal amount of the number of sequences, but an unequal amount in the number of F and M genomes, right? So we know that the female can contribute or have the F genome, right? Whereas the male can have both that as well as the M genome.
Yeah.
So it would make sense that you would have more F genome because both the female and male can hold that F genome.
Ah, I see what you're saying. Okay. I see what
But
you're saying. That makes
since
sense.
the male can only hold since the male genome can only be held by the male, it would make sense for there to be a smaller amount of that, right? It's
Yeah.
almost like for the female, like 20 sequences will be, I believe, 20 of the F. For the male, however, 10 will be for the F and 10 will be for the M.
Okay.
So the 20 could account for the 20 here in the 30, but the 10 to go from 20 to 30 will come from the male. And then the 10 here comes just from the male, right?
Yeah,
So
that makes sense.
I think that's, yeah. And like nothing really indicated to me like, well, so the reason I looked at because like I wasn't really sure what they meant by number of sequences, right? Um, but I noticed that sequence length is the same, polymorphic sites kind of similar, and number of haplotypes kind of similar. Um, and uh, from passage information, we know, this thing over here. And also the answer choices help us out because they're all talking about number of sequences, so that kind of helps us with where to look.
Okay,
So
that makes sense. The estimated number of sequences in the genome is less than that of less than for males. Okay.
yeah, so B is contradicted by the passage table because we see that the number of sequences from the F genome is greater than the than for males, right?
Yeah,
So
it's 30
B,
versus 20.
yep, and so B is out, and then C says the number of sequences from the F genome is greater than that of the M genome, which is true, but that would support the conclusion that certain mitochondria are heteroplasmic. So this is wrong. Oops. So this is wrong because it says less. This second part is right, but it's wrong because it says no. And then D, the estimated number of sequences from the M genome is less than, wait, the number of sequences from the M genome is less than from females. I don't even know what that means grammatically. Um,
So we know females only have the F genome, so that wouldn't be a correct
yeah,
way
yeah.
to compare. Okay. I
God. Yep. Okay.
mean, that makes much more sense.
Hmm.
I guess we can do this too, to be honest. We can do all the questions here. Researchers noted that the M genome was transiently localized in the gonads of male P. preparatus. What explanation best accounts for the observation?
Okay. So what, yeah, tell me about this question, Stem.
So essentially they're saying that the M genome is only in the gonads of the males. So why is it possible?
What
Why is this
what do you
possible?
mean by males?
The in the gonads of the males.
Oh, Okay. All right. Yeah, I guess I looked at that different because I thought when they said gonads that because they said in the
All
passage
right.
that
Yeah.
for the female it's in the gonads, for the male it's in the mantle. But sure,
Yeah.
okay. Let's say, oh, transiently localized. What's transiently mean?
Transition, like
Transient?
transient,
Yeah.
like it's transition. That's what I'm thinking. Like it's
Like
fading.
temporary.
Oh, okay. Temporary.
So the M genome was temporarily localized in the gonads of the male. And that would make sense because they told us that it's not found in the gonads of the male, it's found in the gonads of the female. So what would make, account for this observation, right?
What would account for the observation? That was why it's temporarily there. Um, so occurs for both F, no, sorry, it could occurs for both M and F genomes. Well, the question sim is only talking about M genomes, so I don't know why it's talking about F. So I don't know if that, I feel like we can cross that out because of that. It's not focused on the M genome.
Sure.
Is independent of sorry, it's independent of M genome function. I feel the passage doesn't really indicate any information about the functioning of the genome. It's kind of just talking more about how it's passed down.
Okay.
Occurs only occurs only for M genome containing mitochondria. We know that doesn't make sense because the males actually have both F and M genomes.
Okay.
So I guess yeah, we're just left with D. It's necessary for transmission of the M genome.
Yeah, so what we're being asked here is why the M genome is normally like localized in the mantle. So, but if you found some in the gonads, why, like temporarily in the gonads, why would they go there, right? And I think it would perhaps require some type of, I don't want to say outside information, but like a gonad is like a sex organ, right?
Mm-hmm.
So it can possibly go there to be transferred like sexually
Oh,
to
yeah.
uh you know for the next generation of kids or whatever.
Okay, that makes more sense. Okay, gotcha. Was my reasoning correct for the other ones? Like, why do you think the other ones are wrong? It's just like A is because it's not talking about it. It's B is not because it's not really focused on function, and C is just
Yeah, so let me see. So A, so they're asking us why did we find the M genome in the gonads of the male? So if you said that it occurs for both M and F genomes, like if that was the case, like why is it that for the female that can have uh let's see, the F genome localized in the gonads, transmitted only by females to offspring of both sexes. So yeah, I guess like yeah, having the F genome shouldn't affect why its location would be someplace. And also like, whether it's true or not, like it doesn't explain why it would be located there. Um, like let's suppose that the F genome can also be transiently located in the gonads of the male. That even if that was true, that wouldn't explain why it would be there, right? And then B is independent of M genome function. Now that can also be true, right? But whatever the M genome's function is shouldn't have anything to do with where it's located, right?
Yeah.
And then C occurs only for M genome containing mitochondria. Um, I mean, so that's saying that this only occurs in, I guess, perhaps males, which I guess could be a little better than A and B because they are saying that, this is in the male. Um, but it still doesn't really answer the question. It's like
Mhm.
saying like you know, why is a male's, whatever? Why does a male produce sperm? And the answer, if your answer is this only occurs for males, right? It doesn't really answer the question. So,
You see what you're saying?
yeah. So is necessary for the transmission. That could be the only explanation about why it's located where it's located. Yeah.
Okay. Combined with the findings shown in Figure 1, the results of what additional test, if performed, okay, so what additional test can we do? It be most helpful in validating the hypothesis that the F genome is evolutionarily more ancient than the M genome.
Ah,
So I remember when I was doing this, I was between A and C.
yep.
I took out D, introducing the genomes into viral vectors. I don't really know what that would do. I mean, yeah, it didn't really make any sense because you're just checking for you're more so checking how the genes are, like more about the genes. Digesting the genomes with site-specific nucleases. If you're just breaking that down, I don't know what that would do.
So let's take a look at figure one again, right? What do they do here?
They're checking for the frequency of mutations.
Yeah, right. So,
Okay. I
so
mean, the thing is with C, it's saying measuring the relative mutation rate. I mean, it says
huh?
what additional test? Aren't they already doing that right there? So,
Oh, so okay. In here, they're saying to detect differences in the number of mutations accumulated over time between sample types, and thus to establish if an apparent difference exists in the age of each genome, an analysis of expected versus observed mutation frequency was also conducted. So, so we have, let's see, so we have our white bars as the actual frequency observed, and the like curve, the line, is the expected um mutation frequency, right?
mhm,
So, but they're still separate, meaning genome M is, the one on the left, genome F is the one on the right, and we can't make a direct comparison between the two because what we see in figure one so far is just, the mutation rate and how that's actually there compared to what is expected to be there. But if we're trying to figure out, in addition to using figure one, what additional tests that we can perform to figure out if one is older than the other, right? So that means we would have to compare the two, meaning compare the, genome M, the genome F,
Makes sense. Because again, so as you're saying, to recap, like that figure one, it doesn't really um it doesn't give us a good understanding. It kind of just compares two different things.
Yeah, so each of these compares just what is observed compared to what is expected.
Okay.
But we can't have a direct comparison between the two, right?
Okay. I see what you're saying.
So if we look at these choices here, we can see, okay, inducing mutation at polymorphic sites within the genomes. Um, so you're causing a mutation there, but that won't help us figure out if one genome is older than the other one, right?
If one genome is older than the other one. Yeah.
Like you don't want to do anything to the genome. Like if you're trying to figure out how old it is, you can you can count, I
Mhm.
guess, the number of like mutations that happened, but
Mhm.
that would be like counting or measuring something rather than doing something to it.
I see the difference.
Hmm.
Like, what if A said, like, what do you think if A said measuring then? Measuring mutations in poly, does that change anything or no?
Yeah, yeah, yep, that would work. So like, yeah, measuring mutation at polymorphic sites within the genome, which is what they do, right? But I think the only thing that you would want to add that C has is relative, right? Relative meaning
Compared to them.
comparing, yeah, genome M to genome
Mhm.
F. So, okay, we
Okay.
can get rid of A because we're doing something to it. We can get
Sure.
rid of B because we're doing something to it. And then also we can get rid of D because we're doing something to that. Uh,
I see what you're saying.
C is the only one that says to measure.
Okay.
Mhm.
See that. Okay. Yeah, this was easy. Yeah, this was kind of more of just
Oh, yeah, yeah, yeah.
one thing, which I think
Nice.
we can go over this. I was kind of just like, I remember I looked at it in Site C, and I'm like, wait a second. I think Site C only carries one electron.
Uh-huh.
So if you want to make two molecules of water, so I was like, okay, maybe that's four, but that was kind of just, I don't know. I don't know if that's just the extent to it.
Yeah, right. Because if you did that like balanced equation or whatever, it would be like one half O2 or one half O2 for like one oxygen, and then it's like forgot what it was for water, but I think that let's see, one electron for each from each of the four soluble cytochrome C, transferring them to a single water molecule, I'm sorry, single oxygen molecule, and thus converting, oh yeah, because this would turn into like the H2O. So
Yeah.
if we have two of these, wait, they're asking us transfer of electrons from how many molecules of cytochrome C in order to catalyze the formation of two molecules of water for molecular oxygen. Okay. Transferring them to a single oxygen molecule and thus converting one O2 into two molecules of water. Yeah, I guess just, this over here. Like I forget the exact balance reaction for like glucose, which you can figure out. Like it's not really a hard stoichiometric thing, but like glucose, it would be like C6H12O6 plus O2 yields CO2 plus H2O. Now, since we have six carbons on the left, we should have six CO2s here. And then
Yeah.
we have eight oxygens on the left, and then on the right we have six times two,
13,
12,
12, and then 13 from H2O.
13 from H2O, but on the left we have wait, eight. So I'm gonna have to change the number of oxygens too. So okay, if we have okay, 12 hydrogens on the left That means this is going to have a 6 on it for 12 hydrogens, right? And so on the right, we have 6 times 2 or 12 plus 6 or 18 oxygens.
Wait, isn't glucose in the phrase of O2? There's not six oxygens, right?
C6H12O.
Two, right?
No, it has 6 oxygens, right?
Oh, that's right. Sorry. Yeah, you're right. You're
Uh-huh.
right. You're right. That's right.
So, yeah, okay, what was I doing here? So, um,
So we have 12 and then 18.
6, 8,
So we just added, I think we added six here, right? To the,
12, then 6, 18, 12. So 6 over here, I believe. Is this it? 12 plus 6, 18 over here. 12 plus 6 is 18. Okay, I think this might be it. Um, okay, yeah, that should work. But one oxygen molecule into two. Okay, that's different here. Um, we have oh, okay, okay. So it's 1/2 O2 plus 2 electrons yields, and I guess plus H plus or 2 H pluses yields your H2O.
okay.
So, so okay, if they're saying 2 molecules of water, we would need 1 molecule of O2, and for each molecule of oxygen, you use 2 electrons. So, like, you know, 1/2 O2 is just 1 oxygen, right? And then that produces H2O. But if we wanted 2 H2Os, right, we're gonna need 2 oxygens. So we need 2 times that. All right.
Right.
So yeah. So yeah, at the complex four, you have 2 electrons go to oxygen to one oxygen atom to produce one water atom. And so if you're producing 2 molecules of water, you're going to have double the amount of O2, which is going to be instead of 1 oxygen, 2 oxygens, and double the amount of electrons from 2 electrons to 4. Yeah.
Okay. And site C only carries one electron, right? Probably. Think. I mean, because how many molecules of cytochrome C do you need? Okay. See if there's anything else here. This was fine.
Mm, So yeah, I guess so. The reduced cytochrome C in the ETC transfers an electron to compound complex 4 and detaches, and 4 such transfers provide electrons to produce 2 water molecules. Um, the transfer of 4 electrons accomplishes the production of 2 water molecules as the first step of reduction. Also pumps 4 hydrogens out to contribute to ATP production. This process returns the elements of complex 4 to the original states to begin another cycle. So it seems like, you know, I think the cytochrome C has to make like 4 trips because it can only transfer 1 electron at a time, right?
Okay, that makes sense. Okay. Oh, before we go, I want to ask, I mean, we got thinking 30 minutes. But we're continuing, I meant to say. Yeah, so should I, when I actually ended up buying this, I had to buy the last 4 exams here, which kind of sucked, but whatever. But I got another Q bank with it. So, I've heard very iffy things about the Blueprint Q bank. I don't think I tried to look through Reddit, but I was not able to find one good comment about it.
Uh-huh.
But yeah, so should I buy UWorld or should I just do that?
Where are you at in the aamc section banks
Oh yeah, I have like 20 questions left of chem. And
of
then
the first section
yeah,
bank?
I haven't touched the second one.
Okay.
And
Yeah,
then the
let's
pH section.
yeah. Yeah, we can work on that.
Okay.